excited for you guys to see what he's been working on the past few years. As usual, I just want to start with a very thorough introduction of Parker as we celebrate the soon-to-be Master Parker today. So I actually met Parker when he was working, when I first got back and started the job at Moss Landing, and I was working out at Onyo. He was a regular person out there. And he actually was sort of a lab adoptee before he joined the lab. So he worked with a lot of the students for wiener weighing. And when I asked for photos, it's not surprising, there was a lot of pictures of the lab out at Onyo with Parker. And when I saw that he could make the funny faces like everyone else, I started to think he could be good for the lab. So I was very excited when he approached me about being a graduate student. And then really what was kind of the selling factor is I'd always heard that, researchers can start to look like their study animal. And when I saw that he was already really looking like the elephant seal, I thought, man, how could I say no? But then I thought, we'd actually talked about an elephant seal project, but I thought it could be fun to see if he could turn into another species. So when he joined the lab, we plugged him into the emperor penguin project. He was a very important asset in the field. He had some key field skills. He was an excellent cook. Very important, very stylish, as you can see in those photos. Again, very key for when you're living in a hut with 3-4 people. You need to be entertained. Also, when you're sleeping on the ice, it's helpful to be able to sleep anywhere, and he can, even lying out on the ice. He was excellent at photo documentation. This is maybe 1/10 of the selfies he took on the day we were leaving from New Zealand to Scott Base. So this is us walking to the airport there. This is us walking towards security. This is us on the bus after security going to the airplane. This is us getting off the bus and walking to the airplane. And then on the airplane, and now he's in the cockpit, so more airplane. And now we're about to get off the airplane. And then we're off the airplane. And really, this is probably 1/10 of what was on there. And then this is on the bus from the airport to Scott Base. And then this is us walking to Scott Base. So he really was great at documenting pretty much every step of our project, including our meals. I have to admit, I got tired of sorting through the thousands of photos, so this is just a snapshot, but he took a selfie, had us, and I guess it's not a selfie, but had us take a picture of us at dinner every night we were there. So day one, we're happy, smiley faces, still looking pretty good, moving through. I'm probably starting to look a little concerned there. Everyone else is still pretty happy. We all have our days. Another selfie, another dinner, getting rougher and rougher. We haven't showered. We're starting to not look at the camera and just focus on eating. And then this is our last meal. We had some groups, so this is one of our last meals, and we're all starting to be a little haggard. No showers for the men, no shaving. So it was great to have all that photo documentation. Also, it's really important to have entertainment. And so Parker was a great dancer. So one of our main jobs in the field camp was to be melting snow, so we have water. And so here he is, melting snow. Unfortunately, he's listening to headphones, so we can't see the music he's dancing to. But again, like I said, quite entertaining, great to have in camp. But in all seriousness, he was a very hard worker. We had to do scans from 6 in the morning till midnight. The last field season, he held most of the birds. Many of them weren't quite as nice as that one that's just leaning across his leg. I was looking for a photo, I didn't get it in time. But he's a great team member, so he was a member of both the 2019, and then unfortunately we had 2 seasons canceled, but then also a member of the 2022 team. And he took a beating, and I was going to show a picture of that, but I didn't get it on here on time. And he really threw himself into this work, and so at some point I'm going to find this video that shows that, but again, I couldn't find the right hard drive, so unfortunately some of that is missing. And we also did find that after 2 seasons working with emperor penguins, he did start to look like the species he studied. So, and then just kind of last, in addition to being an asset in the field, he was also a very important lab member. When I asked people for photos, apparently bowling was a common theme. But he was always participating in the different lab activities. He helped people, he provided people useful feedback. He's very much a loved member in the lab group. And so this is just some of the group photos and stuff like that. So it's been wonderful having Parker in the lab, and I'm very excited for him to share what he's been working on. Now I will pass it off to Parker.
All right, move this aside. All right, welcome everyone. Thanks so much for that awesome introduction, Gita. Welcome to my defense, titled The Influence of Intrinsic Factors on the At-Sea Behavior of Lake Chickering Emperor Penguins at a spectacular place on the planet called Cape Crozier, which is located on Ross Island, and that's in Antarctica. So I'll be introducing you to that specific location in a little bit, but I first want to start out with an emperor penguin video that we captured in 2019. So this is what their at-sea behavior looks like. This is just a sneak peek of what we'll be talking about shortly. But before we get there, I first want to talk about marine predators in general and what our motivation is for studying them. Marine predators are incredible sentients of change in our global ocean environment, and by studying them, we get this really interesting top-down perspective of what's happening in the ocean as far as what prey dynamics are doing, how prey dynamics are shifting from season to season, and it, by proxy, kind of gives us this overall indication of how the overall ocean health is. And the way that we study them is by instrumenting them with these small data logging devices, these small computers that are incredibly powerful. The technology that's on board is not that dissimilar from what you and I might use on our cell phones or our smartwatches, but allows us really interesting insights into marine predators where we're not able to physically observe them, such as when they're diving deep, such as trailing some of our species like our cetaceans, our pinnipeds, our sea turtles, or following seabirds as they ford some of the stormiest conditions on the planet. Speaking of seabirds, they're one of the most diverse marine predators. There's over 360 species that are found worldwide, and they're primarily central place breeders. And what that means is they annually will haul out on either islands or coastal regions where they will rear their young. But as marine predators, they need to acquire resources from the ocean, so that requires them to head out to the ocean. There's a bit of a commute there. They need to find enough prey for themselves, but also for their young. Foraging in the ocean environment is extremely challenging. It's dynamic, and the survival and success of seabirds hinges on their behavioral adaptability and their ability to find and capture sufficient prey sources for not only themselves, but also for their young. The breeding success of seabirds can be influenced by intrinsic factors, and I'll be talking about this over and over again today. It's intrinsic factors such as their sex, their size, and their body condition. To better understand how further understanding how intrinsic factors influence their behavior is crucial to unveiling how these predators optimize their performance at sea during critical phases in their life, such as when they haul out to molt their feathers or when they're breeding. So first I'm going to be talking about the influence that sex, an individual's sex, can have on seabird behavior. I'm mostly going to be focusing on monomorphic species, so species that are visually similar in the way they appear. This is different from dimorphic species, such as elephant seals, is a really good mammal example. Males are quite a bit larger than females. They have that proboscis, and they're not easily confused with females, which are a lot smaller. So monomorphic species, they're visually similar. And while they're visually similar, they might still have slight morphological differences. So for example, in the upper right corner here, we have an albatross. Some albatross species, males have slightly longer wingspans than females, even though they visually appear to be the same, which allows them to fly with greater ease. And then on the bottom right-hand corner here, we have northern gannets. And northern gannet females are actually slightly larger than males, which allows them to dive to greater depths. So they're able to partition that niche. Females are diving deeper, males are diving a little bit more shallowly. And while I'm talking about the influence that size has on birds' diving abilities, among some of the deepest diving seabirds on the planet are our penguin species. What I have here is examples of some of our Antarctic penguins. From our smaller penguin, the Adelie, they're small, but they're also mighty. They can dive to about 180 meters. Versus our largest penguin, the emperor penguin, which can dive to 565 meters. So the main takeaway from this slide is that as size kind of increases across different species, we start to see birds are able to dive deeper. And the reason that is because larger sized seabirds typically have lower mass-specific metabolic rates, which allow them to utilize oxygen more efficiently or slowly while they're diving to great depths. In addition to that, you'll notice that the flippers, or the flippers on these penguins start to get a little bit longer, which allows them to dive with greater ease. They have lower dominant stroke rate frequencies. They're not stroking as frequently as they dive to greater depths, and they might be more well adapted for deep diving. So the size of an individual can influence their behavior. Larger individuals are able to dive deeper than smaller individuals. Moving on to our last intrinsic factor, it's body condition. So body condition index can definitely influence breeding seabirds' behavior. It's similar to, so body condition index is similar to a body mass index, if you've ever measured your own, and it's an overall indication of a seabird's health, the fitness of the bird. It influences their survival, their stress resistance, their energetic demands, and of course, their reproductive success. Here we have an example of two different emperor penguins. One's in low body condition, one's in high body condition. When breeding, seabirds with low body condition can potentially have longer foraging trips. They might need to travel farther, and this comes at a provisioning cost, as they're spending a greater percent of their time commuting out to sea to find resources to sustain themselves, as well as to bring back for their young. Versus individuals that have high body condition, this larger looking penguin here, they might have shorter foraging trips. They might not need to travel as far from the colony. Perhaps they just need to acquire a few small resources to bring back for their young. This comes potentially at a provisioning benefit to their young. Of course, that's dependent on prey availability. Speaking of prey availability, I want to cover one extrinsic factor. So I've talked already about how sex, size, and body condition can influence seabirds, and that's an intrinsic factor, so how the physical form of a bird can influence their behavior. Now I'm talking about extrinsic factors, so factors that are external, abiotic factors. And I want to introduce you to the Ross Sea where our study takes place. I'll be giving you a better indication of where that is in a second. We'll take a tour. But first I want to show what the bathymetry off of the Antarctic continent looks like on the Ross Sea. We have these shallow, relatively shallow bathymetric features that can be called, they're called bank areas, or you can imagine them as underwater mountains or seamounts. And these are critical areas where, key areas where micronutrients are advected as the circumpolar current comes across the Ross Shelf, interacts with these shallow bathymetric features, kind of advecting micronutrients. A key micronutrient, limiting micronutrient in the Ross Sea is iron, similar to what we've been studying here at Moss Landing in our physical oceanography classes and our chemical oceanography classes, and that stimulates the bottom of the food web. So these regions off the coast, these relatively shallow bathymetric features, are highly productive zones that stimulate the entire food web. As part of this food web, we have our penguin species that are mesopredators. They're foraging on primarily fish and krill, as well as some squid, and their only predators are the killer whale and the leopard seal. So penguin species in general are not doing so well. Of the 9 species that are depicted here, these are subantarctic and Antarctic species. About 5 of them are experiencing losses in their population dynamics. So the emperor penguin, this figure needs to be a little bit updated. There's a question mark next to the emperor penguin, and that's because of last year they finally were designated as a threatened species. And the reason that is because they're what's considered an ice obligate species. An ice obligate species requires seasonal ice formations for key aspects of their life. They need it in order to breed, they need it in order to raise their young, they need it in order to avoid predators, they need it in order to haul out on ice to rest while they're at sea. And as you can imagine, as climate change is starting to impact our polar regions, we're losing sea ice. And sea ice is becoming more unpredictable, which threatens their existence. So currently, emperor penguins have a circumpolar distribution around the Antarctic continent. There's about 600,000 breeding pairs, and these black dots are where their current colonies are located. And under the assumption, this model is predicting that by the year 2100, under the assumption that the climate will warm by 2.6 degrees Celsius, these red regions, we're potentially going to lose about 90% of the population. These yellow are 50, blue is 30%, And these gray areas are gray areas. These are areas of key conservation interests for the species, and that's conveniently also where our study takes place, below this penguin on the Ross Sea here, located there. I don't want this to be like a doom and gloom kind of perspective. Again, this is a model, so if we're able to change our behavior and reduce carbon emissions, potentially we can save penguins, so just keep that in mind. So now, this is where Cape Crozier is located. So this is a similar map as you saw before. Cape Crozier is the second most southern colony in Antarctica. It's the most southern breeding colony on the Ross Sea. And the reason that we're studying them here is because of the potential conservation implications, and their at-sea behavior has never been documented here before. So for the very first time, we're studying them at this critical area. So we'll take a quick tour. We're gonna zoom in to Ross Island. For those of you that have been to McMurdo Station, that's found up here. Across from McMurdo Station is a place called Scott Base. It's run by New Zealand, and that's where all of our funding came through and all of our logistic support came from. So we gathered all of our gear together, put them in helicopter sling loads, and we're helicoptered out to Cape Crozier to live on the ice with the emperor penguins. So we'll be going there right now. All right, so here's where Cape Crozier is located. On the bottom right corner, what you're seeing is this thinner ice here is what's referred to as temporal forming sea ice. So when we're there, there's about 2 meters of ice below our feet. But during January, so about 3 months after we're there, this becomes part of the open ocean. On the far left-hand side, we have this thicker-looking ice, and that's glacial ice. That's called fast ice. It's fasted to the continent, and as it constantly moves towards the continent, it starts to break up and create these fingers in the ice, or canyons. And we'll zoom in a little bit farther. And this is where the penguins like to raise their young. It provides a bit of a thermal barrier. This is one of the most turbid places on the planet, and you can actually see a little bit guano from this old satellite image right here. So that's where Cape Crozier is located. So I'll be going over the breeding cycle of emperor penguins real quick. So they mate in May. A single egg is laid by a female. She loses about 35% of her body mass during that time. She passes that egg off to the male, who incubates that egg for 120 days. The male, she heads out to sea to recuperate her energetic losses. The male, in the meantime, has a really important role to not drop that egg on the ice for more than 6 seconds. Otherwise, that egg will pretty much be unviable. The female will return when the chick hatches at the end of July into August, so coming up here pretty soon here. We'll provision the chick, will help thermoregulate the chick while the male heads out to sea. So over that 120-day incubation period, males lose between 35 to 50% of their body mass, need to head out to sea really quickly, acquire enough resources to bring back for the young. Our study takes place towards the latter end of the breeding cycle, which is referred to the late chick rearing season, when the chicks are able to thermoregulate more or less on their own. Both adults are heading out to sea to acquire food for their young. And this is an extremely stressful time to be a penguin. Chicks are experiencing rapid growth, so they need extra calories. Adults, they have about 3 months to fully develop before the ice beneath their feet melts and they need to successfully fledge, at which point they will spend the first year of their life on their own. So towards the end of December, January, this chick needs to be ready to fend for itself. So now onto my primary objectives. So these are the questions that we seek to answer in our study. I seek to better understand how adults meet the energetic needs of their chicks during this critical energetic phase, better understand whether or not they exhibit any type of strategies while they're at sea, so documenting their behavior, and documenting whether or not there are similarities or differences in their behavior, and lastly, do intrinsic factors such as an individual's sex, size, and body condition influence how they move while they're at sea. Moving on to my hypotheses. So my first hypothesis was very well informed by observations made by Dr. Koyman in a paper published in 2021 on the Quarry Bank. So their study took place up at Cape Washington, which is located to the northwest up here, and it's the largest emperor penguin colony on the Ross Sea. At this location, what they documented is that penguins tended to dive deep over the shallow bathymetric feature, those bank areas that I was referring to before, a place called the Quarry Bank. So similarly, at Cape Crozier, we do have a bank area, but it's about twice the distance of what was previously published. So we didn't know technically if they'd be able to travel that far. So this is the Ross Bank. This is where Cape Crozier is located. Based off of those initial observations, my first prediction was that penguins, if they traveled over these bank regions, they'd be exhibiting similar behavior and that they would dive deep. If penguins were traveling over deeper water, such as off the coast of Cape Crozier, they would be more or less diving more shallowly. Building upon that, if penguins were diving deep, the reason that they're doing so is because there must be a reward for them. Diving deep is extremely taxing, and they're going to be either rewarded with greater quality of prey or quantity of prey. So I predicted that deep divers likely would have deeper average dives. They would exhibit a greater percent of near benthic dives, and they'd have fewer dives per day because diving deep is exhausting, and that they would have longer trip lengths. So I expected them, if they're traveling that far of a distance out to these bank areas, that they'd have longer trips. Versus individuals that were diving more shallowly, I expected them to have shorter trip lengths because they're not going all the way up to the Ross Bank, that they would have more dives per day, that they would have faster surface intervals, and higher stroke rates. So the reason I predicted this is because I expected that prey that's found in shallower water are probably more dispersed. Penguins are likely traveling quicker through these patches and trying to capture prey that are quickly moving. Moving on to my intrinsic factors, predictions. So I'll bring up the first two. They're kind of informed by each other. We'll start with size. I know it's out of order, but larger penguins I expected to potentially be able to dive to greater depths, and I expected males to be slightly larger than females. So males would probably be deeper divers, that they would perform fewer dives because diving deep is exhausting, and that they have a greater percentage of mesopelagic dives, meaning that they're diving deeper than about 200 meters more often than females. Females, I expected that they would dive more frequently, they would be epipelagic foragers, and that they primarily be shallow divers. As far as my body condition index predictions go, I expected individuals in higher body conditions to have shorter trip lengths versus individuals of low body condition. I expected them to have potentially longer trips. Moving on to methods. So the first thing you need to do is capture a penguin. And the way that we did that is by capturing them about 1 kilometer seaward of the colony. And there's constantly a stream of penguins coming and going from the colony, penguins coming back from their trips. And this is kind of what that looks like. So the penguin that's heading towards the camera is heading out to sea, and this potentially would be a good candidate. I'll be showing you a video of what that looks like shortly. But before I get there, I also want to let you know that we take morphological measurements. Once the penguins are captured, we take a flipper length, so tip of the feathers into the armpit, as well as a girth measurement. And then we also weigh the animals by putting them in a small harness, lifting them up on a Pasola scale, reading how much they weigh. So again, this is a monomorphic species, meaning that they're visually similar. And we need to take feather samples in order to sex them. So we take these feather samples, send them out to get sequenced, and that's how we get that sex variable. So now I'll be presenting to you what the capture process looks like. Well, I won't be presenting it, but I'll be showing a video.
It's here on this frozen frontier that scientists are working to monitor emperor penguins. It's a careful operation. They work together to slowly surround each bird and wrap them up in what they call a big hut. Placing a hood over the head to calm the nerves before attaching some bling and advanced GPS tracker.
All right. So that's how we capture penguins. We then get them ready to deploy our data loggers, or our small computer devices. We take these data loggers, we glue them to the surface of the penguin's feathers using Loctite glue. We use a little bit of what's called Tessa tape to provide a little bit of additional structure. We take 2 zip ties, kind of cinch those zip ties around that tag. That tag isn't going anywhere while they're at sea. We also attach a very high frequency transmitter, which is used to basically triangulate the position of the animal when it returns. So these animals will go out to sea, they'll forage, they'll come back, we'll hear a beeping sound from our antennas from these very high frequency transmitters. We'll be able to triangulate its position, recapture the animal, reweigh the animal when we capture them, so we know how much weight they gained over the trip duration. And then we'll be removing these tags by just simply clipping or cutting the zip ties. We cut the little tape that's attached to it, and then the tags just come off, and there's pretty much nothing left on these birds afterwards. And the data that I'm presenting today is from the 2019 season, in which covers about 8 breeding birds. So aboard these data loggers are sophisticated instruments, but not that different from what we use every single day. It allows us to know, answer the question of where are these penguins going. It allows us to reconstruct what their behaviors are from the tri-accelerometer data, and we also know how deep these penguins are diving. I'll be going into this data in a little bit more detail and showing you what I did. So from the GPS data, this is what raw GPS tracks kind of look like from a penguin. There's all these GPS points. We run this through a speed filter to get a track, so we know where these penguins are traveling. Along their tracks, there's these little squiggly lines, these areas. And from these squiggly lines, these are areas where these penguins are spending additional time, which means that they're probably foraging here. And we're able to estimate the scale of these squiggly lines. It's referred to as the scale of the search area or the area-restricted search. And we're able to better understand how much time they spend within these areas foraging, which is when I'm presenting the first passage time analysis, that's where that data kind of comes from. Moving on to the tri-accelerometer data. This data is one of my favorites. I mostly worked with the X and the Z axes of the acceleration data, but from this data, we're able to reconstruct the behavior of these animals where we're not able to physically observe them. From the static and the dynamic component of the acceleration data, we know the body position of the animal as well as whether or not they're moving, and we're able to categorize their behavior into specific behavior. So for example, here is the animal standing and actively moving, which is the walking behavior, lying down, not moving, which is the resting behavior, lying down and actively moving, which would be categorized as the tobogganing behavior, and there it is standing again. So from this information, I'm able to, at a very fine scale, estimate how much time the penguin spent walking while it was at sea versus lying down, tobogganing, or as well as swimming. So that's how I got my time activity budget variables. And just before I move on, all of this was conducted through a package called Ethographer, following Dr. Sakamoto's methods. In addition to that, I also took the acceleration data and to estimate the stroke rates of emperor penguins. So this figure right here, on the x-axis we have time, on the y is the dynamic acceleration component. The way that you can look at that is the amplitude of the stroke signatures. And the peaks of these green peaks are individual strokes. And so I created an algorithm to detect these peaks. And this gets at so for every single dive, I know how many times that penguin stroked, gives us an indication of the energy that they expend per unit time. Moving on to pressure data. So our pressure data is at 1 hertz, so every second I know how deep this penguin is diving while it's at sea. We're able to put that information through DiveMove, a package that allows us to categorize the dives into specific phases. I know if the penguin is in the descent phase, bottom phase, which is the primary foraging phase, the ascent phase, as well as how much time that penguin spent at the surface, in the surface interval, so how much time it spent resting after the dive. So across these different dive phases, I know how much time that penguin spent in each part of that dive. In addition to that, I also categorize dives into specific dive categories. So dives that were less than 200 meters I refer to as epipelagic dives. Dives that are deeper than that are mesopelagic dives. And in addition to that, I also extracted data, the bathymetry data from the raw sea, from the IBCSP version 1.0, to estimate, relative to the bottom of that penguin's dive, how close did it come to the benthic surface. Benthic surface. So if it became within 20% range of the benthos, that means that penguin was exhibiting a benthic dive. And I just want to point out one package that I use for a lot of these figures that I'll be presenting in my results, and that's a MATLAB package called Antarctic Mapping Tools by Green et al. In 2013. It's an incredible package that I use quite often. So yeah, if you see cool visuals, that's where it came from. So I've described all these different types of variables. I'm able to reconstruct emperor penguin behaviors, but what do you do with that information? So in order to answer the question of do penguins exhibit more similar or dissimilar behaviors, I ran the data through a hierarchical cluster analysis. What that allows me to do is, for example, on the x-axis here, we have the tendency of a penguin to lie down, versus on the y-axis, we have the tendency of that penguin to be standing, in that standing behavior. What a hierarchical cluster analysis does is it kind of groups individuals by similar behavior. I'm then able to create a dendrogram. This is similar to like a tree of life. And individuals that are like this, so I categorize these individuals into 2 different categories. On the left-hand side, we have the likely higher likelihood of penguins to be standing versus on the right-hand side, group B, is a higher likelihood of these penguins to be lying down. But what the hierarchical cluster analysis doesn't tell you is whether or not they're statistically different in those behaviors. So in addition to running hierarchical cluster analysis, I also ran robust multivariate analysis of variances using the NPMV package to test whether or not there were significant differences between one group or the other as far as their behavior goes. In addition to that, I also grouped my data by males and females to compare behaviors between those two. So my last independent parameters are size and body condition index. To assess whether or not emperor penguin behavior was influenced by these variables, I ran general additive models using the MGCV package. I set my significance level at a practical scale because my sample size is very small. And when I'm referring to size, I mean the initial mass of the penguin. When I'm referring to body condition index, I mean the mass of the individual at capture divided by the flipper length. So on to our results. So for the very first time, I'm excited to share with the general public where emperor penguins at Cape Crozier traveled. What you're looking at is a looped figure of all the penguins that we captured in 2019. So this is one trip. It's looped over and over again. They're colored. Each color is a different penguin. And in general, we have penguins that are traveling north. We have penguins that are traveling northeast. And we have 2 penguins that traveled west. On average, they spend about 10.6 days at sea, in which they covered 527 kilometers, about 55 kilometers per day. And the max track length came from penguin 5, this little green line right there. It's incredible. It traveled over 1,000 kilometers, which is the equivalent of traveling from pretty much like San Jose State all the way down to Los Angeles and back, just to forage for your chick. It's remarkable, and we'll be investigating that penguin in greater detail in just a little while. I also want to show you this figure where the tracks are not actually moving. On the far right-hand side here, this is the Ross Bank. And yes, to our surprise, indeed, emperor penguins did travel out to the Ross Bank, but that wasn't the only story. Some traveled north, some traveled west. As far as their dive summaries go, penguins primarily dove within the first 200 meters of the water column. They had a fewer percentage of dives that were in the deeper than 200 meters, which was 8%. And I love this figure. On the x-axis, we have the distance along the foraging trip track. So imagine the penguin's leaving at the origin. It's heading out to sea in the middle part of these graphs. And then towards the far right-hand side of the x-axis is the penguin returning to the colony. The black feature is the bathymetry that was below the penguin's track. The y-axis is depth, and the yellow or orange is the penguin's dive. The main takeaway from this figure is that as bathymetry starts to become more shallow, we start to see deeper dives in most of these penguins, but not all of them. On average, penguins dove about 174 times per day. The max dive depth was 456 meters. The max duration was close to 13 minutes. And the mean dive depth was 82 meters. Next, I'll be showing you one of my favorite penguins, penguin 5, that penguin that traveled such a long distance. And don't be fooled by the change in track colors. I just wanted to create a little bit more contrast with this red line. But this, on the left-hand side here, is where the penguin started, and then it traveled out to the Ross Bank. This lighter feature here. The Ross Bank runs between about 700 meters up to about 250 meters in its shallowest areas, and this darker area right here is over 1,000 meters in depth. Now we'll be taking a quick dive with emperor penguins. So this is what their dives look like below the sea surface. These red dots connected to these black lines are the maximum dive duration or maximum dive depth of this penguin. So as it comes into contact with this shallow bathymetry, this penguin's diving extremely deep. But when it's diving over deeper water, over here on the right-hand side, it's not diving as deep. And similar trends were observed from the first passage time analysis. So within the first 25% of these penguins' trips, they're not spending a whole lot of time. They're transiting to a specific area where they start to forage. The figures on the right-hand side here, the only thing you really need to take away is that these lighter features, these yellow, red, orange areas are where these penguins spend significant amounts of time during their trips. Many of these penguins spend a lot of time over the Ross Bank, but that's not the case for all of these penguins. Some penguins foraged over deeper water. The mean area-restricted search for these penguins was about 50 kilometers, and penguins spent about ranging 10 to 50 minutes within that spatial scale. And in general, northbound penguins had larger area-restricted searches than other penguins. Moving on to our time activity budgets. So on the x-axis here, we have the penguin IDs, so their names. On the y-axis, we have the percent that these penguins spent in each of these different behavioral categories. Primarily, penguins spent time in the water. That's not a big surprise. They spent 2/3 of their time in the water. They spent about 1/3 of their time hauled out. When they're diving, they spend primarily their time in the descent part of their diving phase, as well as the ascent, and a little bit less time in the bottom phase. When penguins do haul out, they're primarily hauling out to rest in the lie or stand behavior. They're not really walking or tobogganing all that much. So here's the stroke rate analysis results. On the x-axis, I have the different dive phases. On the y-axis, I have the stroke rate. And this figure is broken into 2 different dive categories. So I have the stroke rate for emperor penguins on epipelagic dives, 0 to 200, 5 to 200 meters, and then mesopelagic dives, deeper dives, on the right-hand side. The average stroke rate was 0.64 hertz. They stroked a little bit quicker in the descent, followed by the bottom, and the ascent was relatively passive. And across these 2 different dive categories, shallow dives versus deeper dives, penguins tended to stroke a little bit quicker on shallower dives, but that wasn't statistically significant. So our behavioral groups. So from the hierarchical cluster analysis, it identified 2 different groups. We have composing 4 individuals in each group, group 1 and group 2. I quickly just want to remind you of my hypotheses. So penguins that are diving deeper are likely to do so over shallow bathymetry versus individuals that are diving more shallowly are typically diving over deeper water. So what does that look like in reference to these different group compositions? So from the ARMANOVA results, group 1 tended to dive deeper on average. These penguins had fewer dives per day. Diving deep is strenuous. They had longer surface rest periods. They traveled farther distances per day to get out to the Ross Bank. One of my predictions was that these penguins would have more benthic use than individuals that were diving more shallowly. That didn't actually turn out to be true. And they had lower stroke rates, which is also interesting, and maybe we can talk about that in the discussion. Versus group 2, these were our shallower divers. So on average, they had shallower mean dives. They had shorter surface rest periods. They had shorter distance that they traveled per day, higher stroke rates, which is also something that was predicted, but they didn't have less benthic use. They statistically had more benthic use, which is interesting because I wasn't expecting that at all. Perhaps we can talk about that if anyone has questions. So generally, across these 2 different groups, group 1 and group 2 differed in their dive behavior. What's interesting is that these penguins, between these 2 groups, they spent about the same amount of time at sea, which I didn't expect. And what about intrinsic factors? What about sex? How did sex influence the composition of these groups? Well, I didn't know this until pretty recently, the beginning of this year. We finally got our sex data back. And it turns out that on the left-hand side, group 1 is primarily males. On the right-hand side, group 2 is primarily females. So here are my hypotheses, or just a quick refresher. My hypotheses for size and sex. So I expected males to be slightly larger than females, and that they would dive deeper. Males might have longer flipper lengths, lower stroke rate frequencies. Females would have shallower dives. They would have more dives per day, and they'd primarily be shallow divers. Well, was this true? Well, females were, on average, shallower divers. They did have more dives, that they dove more frequently per day. It's a prediction that I didn't have, but turns out that they also had longer maximum dive durations, versus males did dive deeper on average. They had statistically longer flipper lengths, and they had lower stroke rates while they were diving. But when we look at where females traveled versus males, it seems like they're using similar habitat, which is also pretty interesting. So while their dive behavior is very distinct, they're foraging in similar areas. Moving on to my general additive model results. So the size of the bird didn't seem to influence didn't have a linear or nonlinear relationship on these behavioral parameters. But body condition did tend to influence the behavior of these birds as far as their average bottom dive depth goes and their post-dive interval, which I'll be showing you in greater detail here. So on the x-axis here, we have the body condition index. Starting at the origin, we have individuals of low body condition going up to higher body condition. And on the y-axis, we have a spleen for the mean bottom dive depth. The way that this can be interpreted is that as you go higher up on the y-axis, we have the tendency of these penguins to dive deeper. Initially, so penguins in lower body condition tended to not dive as deep as penguins in intermediate body condition, and penguins in higher body condition tended to also not dive as deep. This similar trend was also reflected in the amount of time they spent resting at the surface. So all that's changed here is on the y-axis, I have the mean post-dive interval, so the amount of time that they're resting following a dive. So individuals of low body condition are not resting as much as individuals in intermediate condition, and individuals of high body condition are also not resting as much because they're not diving as deeply. So moving on to our discussion. So in general, we now know where penguins at Cape Crozier traveled. They primarily traveled north and northeast. 2 traveled west. We know that they exhibited two different types of behaviors. We have our deep divers, many of which traveled out to the Ross Bank. We have our shallow divers, some of which stayed actually close to the continent and found little places where there was shallow bathymetry and were foraging there. That was also unexpected. We also had some individuals that were traveling directly north and foraging over deeper water. In general, the trend does seem to be true that penguins tend to dive deeper over these shallow features. So these are features that are really important for primary productivity and kind of cascading up the trophic food web. And speaking of that, I just want to show you where some of these emperor penguin prey are actually located. So in this box, this small little island there is Ross Island. Off the coast here, we have one of emperor penguin's favorite prey sources, which is the Antarctic krill. So they're quite abundant where these penguins are foraging, as well as it's found out on the Ross Bank as well. Another prey type is the crystal krill. So they're primarily found a little bit farther off the coast, off of the Ross Bank. And then we have one of their favorite prey, which is the Antarctic silverfish. And I think this one's actually pretty interesting. So they're primarily found over the Ross Bank as well as north. So that might explain why some of these penguins were actually foraging over deeper water. Potentially they're targeting different prey types. Moving on to the influence that sex has on males and female, or sex has on these different types of penguin behaviors. The reason that this might be is likely due to differences in energetic requirements, prey preferences, or physical characteristics. We now know that penguins, males tend to have slightly longer flipper lengths. And the whole reason behind this is that initially when I was talking about the breeding cycle and males have that 120-day incubation period, they have a really short time in which they need to head out to sea to acquire prey to bring back for their young. And I think there's some ecological pressure there where they need to acquire prey resources really quickly. They've lost a huge proportion of their body mass, and what's the best way to do that? Well, it's likely to dive deep. So these penguins might be diving deep. The males are likely diving deeper because they have to. Males also have higher mortality risks than females, so diving deep is actually a risky behavior. So that's kind of my perspective on why males are diving deeper than females. Moving on to body condition, I think these trends make sense. Individuals that are of low body condition, they might not be able to dive deep. They might not have the energetic stores to do so. Individuals that are in intermediate body condition, they might have the energetic stores to dive deep. They might also want to increase their body condition higher. They might be willing to risk it more. Versus individuals of high body condition, why would they dive deep if they don't need to? Perhaps they're just heading out to sea to acquire a few prey items to bring back for their young. So where does this study kind of fit into what we already know about penguins on the Ross Sea? Well, when we compared the time activity budgets that were conducted at Cape Washington, the larger colony with a bank area that was located about half the distance, the time activity budgets between these two locations didn't differ all that much. But I think there's a lot of potential for future studies to be made looking at emperor penguin behavior at these two colonies kind of simultaneously. But in general, the time activity budget doesn't seem to be different between these two areas, and that's likely because all these penguins have similar constraints. They need to get back to their chicks. They need to be efficient, they need to acquire prey, they need to bring prey back for their chicks. Moving on to the conclusion. So we know that the sex of individuals as well as their body condition can definitely influence their behavior. We know that this is interesting in the face of climate models. I think this is going to be pretty important. When we're looking at warming seas, I think that climate change potentially will have variable influences depending on the sex. It's definitely going to, sex of individuals, changing prey composition, and as far as conservation efforts go, Cape Crozier is a critical habitat for emperor penguins. There's the distant Ross Bank that provides incredible resources for them and is worth protecting moving forward. So conservation efforts such as the Ross Sea Region Marine Protective Area is essential. And this is a really exciting time to be doing research with emperor penguins on the Ross Sea. And a lot of the research or future research interests are actually happening right here, which is a statement in itself. And in our lab, Taylor is actually going to be quantifying prey capture events, so figuring out how many prey items these penguins are capturing while they're at sea, potentially looking at differences across these different strategies. And Katie Krager in our lab is looking at modeling habitats. So we know that bathymetry is a key feature, but what about ice conditions? And Gita is also studying them during the molting period, another critical energetic phase of their lives. So it's a really exciting time to be doing research with emperor penguins on the Ross Sea. And a lot of that research is happening right here in Moss Landing. So thanks for listening. That's kind of the end of my presentation, almost. I first have a bunch of acknowledgments that I'd like to go through, if you don't mind holding your applause. First, thanks to all the penguins for inspiring this work. This is a species that's worth conserving. It's worth changing your behaviors to, try to reduce your carbon emissions as best as you can. Keep them around as long as possible. Trust me, they're worth it. And Dr. Gita McDonald, I've had so many incredible experiences with you. Thank you for taking me into your lab. And you've made me a better writer. You've made me a better scientist. And I will forever be grateful for that, especially that one time that you saved my toes when I got frostbite. Yeah, I wouldn't be literally standing here without you. So thank you for that. And Dr. Thomas Connolly, to be honest, I felt pretty out of place here when I came to Moss Landing, and your physical oceanography class really inspired me to try harder. Coming to your office hours, pretty much I was there every single day, and I don't think you know how much that meant to me. You continually made me feel like as long as I tried hard enough that I deserved to be here at Moss Landing, but also deserved to be here in science. So thank you so much for being that incredible motivating factor, and thank you for your help with my thesis. Moving on to Dr. Kim Geets. Your passion for science is just infectious, and you ask such incredible questions, and it's shaped my thesis in ways that you improved my thesis so much. Thank you so much for all of your edits, and all the figures that I've made in this thesis were kind of inspired by the figures that you've published. So thank you for being such an incredible scientist, and thank you for asking such great questions. Moving on to our field team. So Dr. Marcus Horning was and will always be the best chef at Cape Crozier. I've learned so much by watching you do science, and every single conversation we've had has been really enlightening, and I would love to work with you again. Thank you, Marcus Horning. Moving on to Dr. David Thompson. This was my tent mate. We've seen each other at our worst when we're freezing. It was minus 40 degrees, and you let me get into my sleeping bag before you did, which was really appreciated. You're an incredible scientist. You have magical humor. I've learned so much by just having conversations with you. Thank you so much, David. Really appreciate it. Moving on to Dr. Katie Krager. Thanks so much for helping me along with my general additive models. I came to you, and I didn't know if what I was doing was the right thing, but you really positively reinforced that I had the skill set to do this work, and you told me that I was on the right track, and I really needed to hear that. Thank you for being one of the most fun people to work with in the field. I don't think I've ever laughed so hard in my entire life. Yeah, thank you for being you. Moving on to our logistics support at Scott Base. So all of these people on this list were essential for our 2019 field season, everything from gathering our gear together to providing food for us to making sure that we were safe and testing the ice. So this is thank you to the 2019 field crew at Antarctica, New Zealand. I didn't present any 2022 data today, but I'd also like to thank the individuals that helped us this last season, especially the people that are underlined. We have Eric, we have Philly, Chris, we have Raf. Thanks to all these people on this list. It takes a lot of effort to get out to Cape Crozier. We take a helicopter, and every single person on this list was essential for that work to happen. Moving on to the vertebrate ecology lab. This is such a great community. It's one thing to like love the work that you're doing, but it's a complete different thing when you also love the people that you work with. Thank you so much for being there all these years. And yeah, the Moss Landing Marine staff and faculty, not everyone is on this list. I apologize for that, but I want to give special shout outs to Tara. Thank you so much for answering my emails and making me feel like I was okay. Thank you to Michelle Keefe for helping with reimbursements, as well as Jane Webster. Thank you, Dr. Jim Harvey. Thank you, Katie, for helping me with references. And of course, everyone else at this lovely university. Thank you to the Moss Landing student body. Thanks specifically to the 2018 cohort as well. And on to my family. Thanks to my mom and dad for being there for highs and lows. They're such an incredible support network. Thank you to my grandpa, grandma, Susie, Dylan, Mark, Doug, the Forman family. Thanks to Nani, Marika, and Justin, Ian and Gwen from coming from Switzerland to be here today. Hi. Uncle Bobby, Patrick and Trisha, Maggie and Sadie, Marta and Kareem, Rue, Taki, and Siri. Thanks to the Nasser family, specifically Claire for your support. Thank you to Tom for being one of the only people to sit down with me and help explain things that were beyond my comprehension. Thanks for being such a good friend. Thank you, Diana. Thank you, Julie, Tony, Moby, and Lenny. And not all my friends are on this list, but thank you to my friends. I apologize. I promise I'll thank you in person. And last but not least, this type of work takes a lot of funding. And so I'd like to acknowledge our funding agencies, the New Zealand Ministry of Business. Thank you for your support. Thank you, National Geographic Society, SeaWorld Conservation Fund, Antarctic New Zealand, San Jose State University, and the RISCA program for their fellowship. Thank you, National Science Foundation. Thank you to my university, CSUMB, and Moss Landing Marine Labs, of course. And with that, I'll take any questions.
Great presentation already, but I was just wondering, did you look at any differences between day and night diving, and specifically related to like sex and depth? Like, was there any differences that stood out if you looked at that, and like related to prey type, maybe like vertical migration stuff?
That's a really good question, and there's 24 hours of sunlight during that part of the season, so it's a little bit different than here, but that is a really good question. I'd like to look at that over different seasons and see what they're diving, how that changes as far as, they're thought to be a visual predator, so I'm sure that light probably doesn't travel that deep, but I'm sure, if you looked at that across different seasons, you might see some differences in their diving behavior.
Has there been, follow-up question, has there been any like isotope stuff that looks at like prey type dependent, like related to sex, or how much has been done?
I don't know if it's related to sex, but there is one paper that has looked at it across different colonies to see if there's differences in composition, and there doesn't seem to be, which is interesting because these penguins, at least at Cape Crozier, seem to be going in kind of different directions, and not all prey types are going to be found by going directly north. So I would suspect that moving forward, research that we're doing in our lab is actually looking at some isotope stuff, it surprised me that there wasn't that much of a difference in prey composition as far as, I can't remember what that study was called, but
Cool.
Great talk.
Thank you.
I have a question about predator avoidance. And if there's any pattern that you could detect in how they were moving or how far they were moving and how that might tie into prey,
Yeah,
seeking prey or the dive behaviors.
so you're talking about like predators like orcas or leopard
Mhm.
seals kind
Or
of chasing them?
Yeah, I mean, is that why they go so far to avoid them? Is there any little movements you can pick up on that might throw off their, or affect how far they're going, or?
That's a really interesting question. Maybe that's why Penguin 5 traveled so far, not because it, was trying to impress us, but because it was scared. I didn't really look at that specifically, but I'm sure you could potentially pick it up in the acceleration data. You could also look at, they haul out on ice when they're scared. Maybe you've seen videos of orcas kind of circling around either seals or, so yeah, as far as predator avoidance goes, potentially you could pick it up by looking at the percentage of time they're spending hauled out, as well as their acceleration signals. But I didn't really look into that, but that's a really interesting question.
I have 2 questions.
Okay.
The first is data driven a bit. So you, when you said that the males, well, I thought it was really interesting that this is a monomorphic species, but then there could be subtle differences, and you got this sexual difference in sexes with their activity. Did you have any thoughts looking back at the data that there might be slight dimorphisms in things like you mentioned the flipper length,
Yeah.
which could affect stroke rate, or like body condition? Could there be aspect ratio differences that if you could go back and sex them and measure, you think that would start to play out?
Yeah, so initially we were actually taking bill measurements as well. We didn't do that this last season, but initial masses for males, as far as body condition goes, was slightly higher, which is interesting, so potentially that might be a factor. Yeah, I'm not sure if that answered your question, but yeah.
Well, I think it could be an interesting thing to kind of follow up on, is asking now if there are slight dimorphic differences, could it be that their behaviors are related to sex or to these subtle differences in there? Yeah.
That's interesting.
So.
Well, definitely flipper length.
Yeah.
But yeah, I'd like to look at that.
My other question is a little bit more imagination based. Okay, this was super cool. I learned so much, and I loved your visualizations and the ways that you approach the data. And you did so much with a logger that had 3 different, logger things on it, right? So if you could design a logger that would expand your ability to follow up on any of the questions that you came out from this, what would be the essential, like would it be heart rate, body temperature? I don't know. What would be the essential thing that you would add to record from?
I think in our field, like heart rate is kind of like the ultimate data. So if you could add a heart rate data and look at what that looks like as far as when they're diving, I would love to look at that type of information. I think video camera data is also like useful as far as looking, kind of documenting. They do this with whales, but they put these little video loggers on them, and they're actually able to estimate ice conditions that these animals are encountering. So I think that could potentially be something that will be added in the future. That the water is so cold and the battery is limited, and we have to keep these tanks to be really, really small. But as technological advances kind of improve, I think heart rate would be wonderful, and potentially adding video as well. Yeah, thank you for the question.
I had a question. You showed really nicely that they dive deeper or shallower leisures, and I was wondering if in the data you can see them doing kind of test dives in deeper water to see if they're over a bank, or does it seem like they have a good sense of where they are?
Yeah, I haven't really looked into that all that much, but it seems like, all of them are kind of transiting within that first 25% of their dive. So I assume that we're catching them towards the end of the season, and I'm sure that they had some test dives earlier in the season to look where prey was. But yeah, I think they must hone in on specific areas earlier on previous trips and use that to kind of inform their decision making on where they're traveling. And I'm not not sure exactly how they sense that, whether there's a chemical cue or what kind of, they're probably not able to see the bottom, but they seem to kind of be very in tune with the shallow bathymetry and where prey is located. Thank you for the question. Jack?
I'm sorry. How does BCI relate to like buoyancy and like the higher BCI animals? Like, you think shallow dives because it's harder to get down deep, or?
I love that. I love that question. Thank you for asking that.
I was waiting for Mason to ask it.
Yeah, I definitely think that plays into it. So I talked about how individuals with low body condition, they might not have the energy to dive deep. So potentially there's this like optimal like body condition that allows you to dive deep before you become a little bit, too buoyant to dive deep. But yeah, that's a really interesting question. I'm not sure exactly how the physics of that go, but yeah, individuals with higher body mass probably have higher fat, and maybe there's a little bit up to a certain point. In the water column, they might have a little bit more resistance. But yeah, I think Mason probably might be able to answer that a little bit.
You have to do some math, I think.
Yeah.
Yeah.
Yeah, that's a good question too. Thank you.
Looks like we've exhausted the audience questions. Are there any on Zoom? I think it's good.
Feel free to email me too if you have any questions.
That computer. All right. Oh, you're connected? Okay, let's see. Where are we? Okay, so why do you suggest that deep diving is exhausting? Deep divers are very careful and deliberate. Shallow diving can be faster and more energetic. And that's from Dan Costa.
Of course.
Of course, yeah.
That's a good perspective to have. Well, I guess my answer to that is then why don't they all dive deep if there's this high prey reward? I think when looking at like emperor penguins and these different types of strategies, the way that I perceive it is that they all have the same goal. They want to acquire enough prey to bring back for their young, and there's many ways to do that. So I'm not sure, yeah, perhaps I overemphasize that diving deep is strenuous activity, but there are greater risks involved in that, and there are greater periods. Also from the prey perspective, as you're diving deeper to find prey, you're putting a lot of energy and hoping that you're going to find prey at depth. You're also kind of losing out on other potential prey as you're going to have to spend a greater percentage of your time resting at the surface. Yeah, that's a good question. I'm not sure exactly how to answer that, but maybe that suffices. Thank you, Dan.
And another question from Luis. Excellent job, Parker. I was curious to know what your thoughts were about individual switching strategies. Do you think they stick to the clusters you found, or do you think they have plasticity to move back and forth depending on the year?
Yeah, I think they definitely have plasticity. That's kind of got a sneak peek at the 2022 data. But across different seabirds, like some seabirds will actually go on a really long foraging trip followed by a kind of shorter foraging trip. That hasn't been necessarily proven yet in emperor penguins, but I suspect that that could potentially be the case with this species as well. So I think they're flexible in the way that they approach their finding prey. But yeah, I kind of cheated and saw the 22 data. We captured an individual that made 2 trips, and it foraged in 2 different types of ways. So there's I haven't looked at its diving behavior specifically, but we know that they're able to go to different locations. Maybe the area that they traveled to wasn't as successful as they had hoped, and they decided to follow a different penguin in a different direction. But yeah, great question, Lisa.
All right, and then a question from Carly Bryce Shadu. If males and females are using the same habitats in different ways, could males and females be eating different prey species? Or since the females and males go out to forage for their chicks at differing time points, could they be looking for nutrient-specific prey depending on the developmental period of their chick?
Yeah. Hi, Carly. That's also a good question. I think it might not be different different prey types. Maybe my hypothesis there is wrong, but I think they're targeting like different quality of prey potentially. Individuals that are diving deeper are probably capturing larger fish versus maybe smaller fish that are more easily accessible at the surface. Yeah, that's my answer to that question. Thanks, Carly.
From Guy Oliver, hi Parker, wonderful job, great graphics. One question, how much BMI change was there between departure and arrival? And also, was there a difference between males and females?
Yeah, so I didn't really include that information in this presentation, but penguins in general gained about, I think it was the equivalent of like 2 hamburgers worth of weight while they were at sea, but I didn't look at differences across males and females. That data came really late in the season. I got this last January or February, and have just kind of started looking into that. That's a really good question, Guy. Thank you.
And you have a lot of compliments that you should read. So I'm trying to find the questions.
That's good.
But great
People like it.
job.
Okay.
Like,
That's good.
that's the, we're trying to, I think that might be, it looked like some of their prey species were denser near Cape Washington. Why wouldn't they head there?
Why wouldn't they head there? Well, maybe it's crowded there. Cape Washington, there's its largest colony. Yeah, why wouldn't they head there? I don't know. That's a really good question.
It would be far. Like, there's already a colony there.
I was interpreting the question as like, why don't they just breed there?
Oh, yeah.
At Cape Washington, but yeah. Thank you.
Sort of on the back end of that, how do they find their prey? Is it chemosensing? Like, do you know?
I don't think that's really known,
Sure.
but probably.
Yeah.
All right. Well, I think we've grilled Parker.